So we are on our last section now for Chapter 44, Osmoregulation and Excretion, and now we're going to talk about the structure and function of the nephron, and the nephron is the functional unit in the kidney. So this is a diagram of a nephron, and the nephron is really sort of the place where the circulatory system and the kidney interact. So these are very small, and you'll see how they sort of fit in with the kidney in just a minute. But let's look at what the circulatory system is doing first. So here we have oxygenated blood. This blood is coming to the kidney from the heart. And even though I might have said that there really aren't two capillary beds that blood enters before it goes back to the heart, within the kidney, it actually does have two capillary beds, but it doesn't travel from one organ to the next. All of that happens within the kidney. So what we have here is we have an arteriole that is carrying blood into this structure right here called the Bowman's capsule. So the Bowman's capsule is basically connected to these tubules, and it's going to be collecting all the filtrate that's coming out of the blood. So you've got this capillary bed right here, and the capillary bed is called the glomerulus. Okay, so that's what the capillary bed is called, and it is inside this cup-shaped tube called Bowman's capsule. So what happens is a bunch of stuff gets pushed out of this capillary bed of the glomerulus into the Bowman's capsule, and then it's going to travel down these tubes. Okay, and these tubes are eventually going to result in a filtrate that we can excrete from the body down here. But there's a lot of stuff that gets pushed out of the glomerulus into the Bowman's capsule that the body needs to reclaim. We're going to talk about the specifics of that later. So this tubule is very sinuous and has a loop in it. It's long. There's a lot of surface area. As you can see, it's also surrounded by another capillary bed. So some of the blood comes in here. Whatever doesn't get pushed out of the glomerulus into the Bowman's capsule, the blood that doesn't get pushed out, or the materials in the capillaries that don't get pushed out, return here into this other arteriole, and then that spreads out into another capillary bed called the peritubular capillaries, which you can think of as surrounding the tubes, peritubular capillaries, and those are going to reclaim some of the material that got pushed out of the glomerulus up here in the Bowman's capsule. Okay, so then after those blood vessels reclaim a lot of the materials that were pushed out, but leaving the nitrogenous waste and other things that need to be excreted, that blood is going to return through a venule and then back to the renal vein, and then this is going to get pumped back to the heart and then back to the lungs. Meanwhile, the filtrate that has now been sent through these tubes, with a lot of the material being reclaimed, is going to be excreted out through this collecting duct right here. And the collecting duct is going to go into the renal pelvis, and where urine is going to be collected. And then from the renal pelvis, it will go to the ureter, the bladder, and out the urethra. We're going to talk about this in a second, but I just wanted to show you this loop that I was talking about. It's called the loop of Henle, and there's a descending limb and an ascending limb, and different things happen in both of those two structures. Again, blood flows to the kidneys and the renal artery. It branches many times to form capillary beds, so there's lots of these glomeruli all over the place in each nephron, and there's thousands and thousands of nephrons. The fluid that doesn't leave the glomerulus is going to go into the this other artery, which is going to form the peritubular capillaries. So here is a quick animation that tells you some of this in a little bit more detail.
The outer part of the kidney is called the cortex. Inside the cortex is the medulla. Urine collects in the renal pelvis and drains from the kidney through a tube called the ureter. A tiny branch of the renal artery carries blood to each nephron for processing. A tiny vein carries blood away. Blood pressure forces fluid through the walls of a cluster of capillaries called the glomerulus, and it collects in the cup-shaped Bowman's capsule. The filtrate is refined as it passes through the twisted nephron tubule. Useful substances are returned to the blood, and the remainder collects as urine. Refined urine is carried by the collecting duct into the renal pelvis for excretion from the body.
So you've seen this figure before, but I'm just going to show it to you one more time because we're going to talk about this in a lot more detail and in a more convoluted diagram in just a minute. But here we have the renal artery that's coming over here. It's bringing blood into this Bowman's capsule here. Here's the glomerulus, is the capillary, and the cup-shaped part right here is the Bowman's capsule. A bunch of materials, and we'll talk about what exactly gets pushed out of these capillaries. They're very leaky in here, and there's high blood pressure, and everything that gets pushed out is called filtrate. And then this capillary is essentially going to stay in close proximity to the excretory tubule, where it can reclaim some of the materials that it lost in the Bowman's capsule. And also further down, it's actually going to secrete some additional materials that need to leave the bloodstream and need to leave the body. So, and then finally, you've got excretion. So these were the 4 parts of excretion: filtration that happens in the glomerulus and Bowman's capsule. Reabsorption will happen along this excretory tubule, and secretion, where additional materials from the blood get moved into the excretory tubule. And finally, excretion. Okay, as I mentioned before, the glomerulus right here, we're going to look at this in a lot more detail. We're going to look at it a little bit more up close. The glomerulus is the capillary bed that came from the renal artery, and it's surrounded by the kidney tubule called the Bowman's capsule. So you can see here how it is totally surrounding those capillaries. Fluid from the blood is going to flow into the Bowman's capsule, and then it's going to get pushed through these capillaries. A lot of material is going to get pushed through these capillaries and enter into the Bowman's capsule. So this figure right here is kind of showing this interface between the capillary and the cells of the Bowman's capsule. You've got this capillary here, what's really leaky. This looks a little bit different from the way we described filtration when we were talking about capillary beds and how the cells are, they've got little breaks between them where this endothelial cells are connecting, but you can still think of it as pores. So lots of material can sort of move from the inside of the capillary to the outside. So as I was mentioning, the blood from the glomerulus is the filtrate from the blood is going to enter the Bowman's capsule. And some of the stuff that enters the Bowman's capsule is water, ions, small nutrients, glucose, all these sorts of things actually leave the blood and go into the kidney tubule. The stuff that stays inside the Bowman's capsule are blood cells and large to medium proteins. So they're going to stay into the blood, and they're going to actually leave and go into the peritubular capillaries, which are going to surround the other tubes of the kidney. So now we want to look at what's going on in these different parts of the tubule. And I know this is a little bit overwhelming, but we're going to go through it a little bit at a time. So here's again the renal artery coming in and branching into these little capillary beds called the glomeruli. So this is one glomerulus, and here's the Bowman's capsule right here. The filtrate is all these materials getting pushed out of the blood and into this tubule. So some of the things that get pushed out are water, salts, bicarbonate, hydrogen ions, urea, which is, remember, the waste product that we make, which requires water to be excreted. Also, nutrients like glucose and amino acids, and some drugs and chemicals. So that all gets pushed out into the filtrate. And then there's a series of different things that happen along the tubule that are going to make sure that the urine that we actually excrete is very concentrated, and that we are able to gain back all of the water that we need and all of the nutrients and other salts and other things that we have pushed out of the glomerulus. We want to retrieve as much of that as we can. So right here we have the proximal tubule, and this happens right after the Bowman's capsule, so the filtrate is coming through here. What this figure is not showing is it's not showing that there are blood vessels. The peritubular capillaries are also lining these tubules. Okay, so whenever you see arrows moving into the tubule, that means that capillaries that are lining out here are either secreting or diffusing materials from the capillary, from the peritubular capillaries into the tubule. And whenever you see arrows of things moving out of these tubules, that means it's moving into the blood. It's moving into the capillaries that are surrounding this peritubular, the excretory tubules. What I want you to notice up here is that the red arrows are representing active transport, and the blue arrows are representing passive transport. So in the proximal tubule, you can see that the tubule is actively transporting salts and nutrients like glucose from the proximal tubule into the blood. And as it's doing that, a few things are following passively. For example, the bicarbonate, ions, water, and potassium are all following passively into the bloodstream from the tubules because of this active pumping. In addition, we have the active movement of hydrogen ions from the capillaries into the tubules, and ammonia is actually going to pass into the tubules passively once those hydrogen ions are moved. So the filtrate now, the blood is reclaiming some of the stuff that it wants, particularly salts and nutrients and water. And then basically what's going to happen is in this is called the descending loop of Henle right here. And what's interesting about the descending loop of Henle is that it is impermeable to salt. So salt can't move in or out of the descending loop of Henle, but water can move. And as the filtrate moves lower and lower and lower into the inner medulla of the kidney, the concentration, you can't see it here, but you'll see it in the next slide, concentration of the interstitial fluid is going to change. Basically what that's going to cause is a lot of water moving from inside of the tubule to outside, and that's where the body's going to reclaim a lot of the water. On the way back up, this ascending loop of Henle is impermeable to water, but it's permeable to salts, and so the body is going to reclaim a bunch of salts, which it needs, through the ascending loop of Henle. And then here's the distal tubule, where there's a lot of active transport here. The blood is losing potassium and hydrogen ions, secreting potassium and hydrogen ions into the tubule. It's pumping out salts and bicarbonates, and water is passing passively. And then what's going to happen is you're going to have a very concentrated urine that's going to be passed out of the nephron through the collecting duct. There's still going to be some reclaiming of salt and water as it passes through the collecting duct. So we're going to talk about these things a little bit more closely, although I've already mentioned this, but now we're going to talk about them from the perspective of the different areas of the of the kidney. So in the cortex, you have both the proximal tubule and the distal tubule. They're on the opposite sides of that loop of Henle I was talking about. You've got the filtrate coming out of the glomerulus. You've got active transport into the capillaries of salt and nutrients like glucose. And you have the active transport of hydrogen ions into the tubule from the capillaries. The passive transport is this bicarbonate, water, and potassium into the capillaries that are surrounding the proximal tubule. And you have the passive transport of ammonia, which is a waste product that's going to be put into the tubule for excretion. Okay, the filtrate's going to go down this loop of Henle, and at the end, it's going to come back up into the distal tubule, again, within the cortex. And there's a lot of active transport here with salts and bicarbonate being actively pumped back into the blood and water following, and potassium and hydrogen ions being pumped from the capillaries and secreted into this tubule. The descending loop of Henle is going to be focused mostly on the passive transport of water back into the capillaries. And the ascending loop of Henle is going to be focused on reabsorbing sodium chloride or salt. And at the lower portion, that salt can be reclaimed passively back into the capillaries, while a little bit higher up, the ascending loop of Henle, the sodium has to be actively pumped back into the capillaries. So that's the outer medulla and the inner medulla of that renal pelvis I was showing you before. In the collecting duct, which is over here, you've got more active transport of salts. So this is the body reclaiming some salts that were in the collecting duct. And additionally, there's some additional reclamation of water and urea, which is weird because, you know, urea is the waste product, but we reclaim a little bit of that, and that's going to be involved in maintaining the osmolarity of the inner medulla, and we'll see that in just a second. So the interesting thing about the nephron and the way it's nestled within the renal pyramid is that there's different concentrations of solutes depending on where you are. The cortex has a lower concentration of solutes, so it's got about 300 milliosmoles, which is very similar to what is in the blood. So this filtrate has a similar osmolarity as to what was inside the capillary and in the interstitial fluid of the cortex. As it moves down, you can see here the osmolarity increases as you move down into the renal, down the renal pelvis into the medulla. And the tubule is basically going to be maintaining much of that same concentration as it moves down. Because of that, as we move down in terms of concentration, that is going to push water out of the tubule back into the capillaries. Okay, so that's why all this water transport is passive. Remember that I mentioned before that this descending loop of Henle was impermeable to salts. So salts can't come in, which would have been the other way of balancing this out. Salts can't come in, so the only thing that can happen is water leaves, and that's going to make this filtrate much more concentrated. Now on the way back up, this part of the loop of Henle is impermeable to water, but permeable to salts. So what you're going to do is, as you are lowering the concentration of the interstitial fluid around here, salts are going to passively leave as you are getting to lower concentrations in the interstitial fluid. This is impermeable to water but permeable to salts. So now the blood vessels, the capillaries, can reclaim salts down here. There's still going to be a little bit of active transport of salts, but a lot of this reclamation of water and sodium chloride are passive, and that is made possible by this concentration gradient that is maintained within the kidney. We have already talked about what's happening up here, specific things that are being reclaimed in the distal tubule. But in the collecting duct, you get the similar sort of effect of this increasing osmolarity within the kidney as the filtrate is moved down. And again, you're going to get this passive movement of water. This, again, is it is impermeable to salts. So there is some active transports of salts, but that's it. You have some urea. Again, I will explain how that urea works in a second, or actually there's a little video, but that's going to help maintain this osmolarity. So here's a few videos that will help repeat this and probably say it in a much more simple way. If nothing else, you'll get some reiteration because it's kind of complicated.
Nephrons regulate the composition of blood by a combination of three processes that transfer materials between the nephron tubules and the capillaries that serve them: filtration, secretion, and reabsorption. A nephron tubule and the blood vessels surrounding it have been straightened and simplified in this diagram. The gradation of color, lighter on top, darker on the bottom, corresponds to an increasing concentration of solutes in the interstitial fluid surrounding the nephron toward the center of the kidney. As blood flows through the glomerulus, a knot of porous capillaries, water and virtually all other molecules small enough are forced by blood pressure out of the glomerulus and into Bowman's capsule. This process is called filtration, and the fluid that accumulates in Bowman's capsule is called filtrate. Blood contains blood cells, plasma proteins, water, sodium chloride, hydrogen ions, bicarbonate ions, urea, glucose, amino acids, and possibly some drugs and poisons. Blood cells and plasma proteins stay in the blood. They are just too big, but all the other substances are filtered from the blood and form the filtrate that accumulates in the nephron tubule. As the filtrate moves along the proximal tubule, certain substances are transported from the blood and added to the filtrate, a process called secretion. Secretion of hydrogen ions helps to regulate body pH. Certain poisons are also secreted to remove them from the blood. This list summarizes substances contained in the filtrate. Filtration is not selective, so some of these substances are useful to the body, and it is important that they be returned to the blood. The kidneys generally need to recover water, sodium chloride, bicarbonate ions, and nutrients such as glucose and amino acids. This reclamation of valuable solutes and water is called reabsorption. Let's look at the process of reabsorption as the filtrate continues its journey through the nephron tubule. In the proximal tubule, reabsorption of bicarbonate ions helps regulate the blood's pH. Sodium chloride, glucose, and amino acids are actively transported out of the filtrate. This leaves the filtrate more dilute than the surrounding interstitial fluid, so water follows by osmosis. These solutes and water re-enter the blood.
Okay, this next video talks a little bit about the loop of Henle and the distal tubule, so you're getting these in little pieces.
As the filtrate moves down the loop of Henle, the concentration of solutes increases in the interstitial fluid surrounding the nephron tubule. Since this portion of the tubule is permeable to water, water leaves by osmosis and is reabsorbed into the blood. This concentrates the filtrate. In the ascending loop, the surrounding fluid becomes more dilute. This portion of the loop is impermeable to water but not sodium chloride. Sodium chloride diffuses out, lowering the solute concentration of the filtrate and adding to the solute concentration of the surrounding fluid. Near the top of the loop, sodium chloride is actively transported out, further diluting the filtrate. More substances are reabsorbed from the distal tubule. Sodium chloride is actively transported out of the filtrate. Bicarbonate ions may be reabsorbed too, helping to regulate body pH. Some drugs and poisons are secreted from the blood into the filtrate at this point, and this is another place where hydrogen ions may also be secreted into the filtrate to further adjust pH. There is a lot going on here. The main point is that the nephron is able to expel unneeded or harmful substances from the blood by filtration and secretion into the filtrate, and the nephron can reabsorb substances useful to the body.
Okay, and this is the last one.
The filtrate enters the collecting duct. As it moves down the collecting duct, it passes through a region where the surrounding fluid has a higher and higher solute concentration. Water leaves the collecting duct by osmosis, concentrating the remaining filtrate into urine. Urea is one of the main body wastes in urine, but here some urea actually diffuses out of the filtrate. The urea performs a useful function by adding to the solute concentration of the interstitial fluid and causing even more water to be reabsorbed from the filtrate. Even though most of the water has been reabsorbed from the filtrate, urine is still 95% water. The most abundant solute is urea, along with other nitrogenous wastes. There is some sodium chloride and traces of other ions such as calcium and potassium. Hydrogen ions make urine acidic, and drugs and toxins may also be present.
Okay, so the important thing to know is that the kidney is complicated. These nephrons, there's thousands of them, millions of them probably within the kidney. All of these are filtering the blood, taking out all of the wastes, reclaiming all of the stuff that we need to keep within our blood vessels and keep circulating around our body. So the kidney is really doing an amazing job of filtering a ton of blood every day. And it's really important in terms of osmoregulation as well. It pushes out so much water in those glomeruli, and then it has to reclaim almost all of that because, as you know, as terrestrial organisms, one of the things we have to be careful of is water regulation. We are sort of prone to water loss, so we need to retain as much water as we can. So in terms of the excretory system, the next step would be once the filtrate, once the urine gathers in this the renal pelvis, this area right here, the collecting ducts all sort of empty into the renal pelvis underneath these renal pyramids. So the urine gathers here, and then it goes into the ureter, travels to the bladder where urine is stored, and then eventually is released and excreted through the urethra. So that's the entirety of the excretory system. The way I presented this initially was that the purpose of the renal system or the excretory system was to eliminate nitrogenous waste from the body, and that is sort of the main gist. But I also want you to get that it's super important and for a lot of other things. For example, regulates blood volume and blood pressure. That it does by determining basically how much water is lost or gained in the kidneys, controls the level of electrolytes and metabolites, regulates blood pH by regulating how much bicarbonate is lost and how much bicarbonate is regained. And most importantly of these, which it's not even written down, but osmoregulation, preserving water, filtering blood while preserving water. It's sort of related to blood volume. Okay. So all of the excretory systems we looked at had a pretty major role in osmoregulation. So just keep that in mind that those two things are closely tied. Okay, so that is the end of that lecture and the end of the lectures for the class.